Eukaryotic chromosomes are linear in shape. In humans, we have 46 total, which makes up 23 pairs. In essence, all of these chromosomes make up all of our DNA, which laid end to end would be 6 feet in length. But this molecule is so thin, it's only 2 nanometers in width. Because of this length, it must be tightly compacted within a microscopic nucleus of a cell. This compaction happens by having DNA spool around special histone proteins. When woven over and over, they create chromatin. But before cellular division, this chromatin becomes more condensed into chromosomes. After replication, the chromosomes decompress back into chromatin. The reason that chromosomes cannot be permanently in a tightly compact linear shape is that it needs to be readily accessible in case a cell needs to build proteins and lipids. Instructions for these are found within the DNA of cells. Chromosomes are copied into pairs called sister chromatids by the time mitosis begins. Each sister chromatid is an identical copy to each other. They are joined together by the centromere region by a kinetochore protein. This kinetochore protein contains attachment sites for spindle microtubules to attach and pull the sister chromatids apart. The cell cycle is a series of steps which explains how a mother cell eventually divides into 2 identical daughter cells. In this chapter, we'll be focusing on the asexual means of reproduction, mitosis, and binary fission. Cells are either in interphase or the mitotic phase. It's also important to point out that in adults, nervous and muscular tissue do not undergo reproduction once matured. Once nervous or muscular tissue is lost, it cannot be replaced. For other cell types, the cell cycle varies as a function of the cell's physiology. For example, skin cells replicate rapidly. Red blood cells last a few weeks before they need replaced, and some cells like memory immune system cells can last decades remembering old diseases. Interphase is when a mother cell carries out normal cell functions, but is also preparing for mitosis by duplicating DNA and cellular components. The vast majority of a cell's life is spent in interphase. Mitosis is when the mother cell begins to divide the DNA and components of these cells, and finally, cells undergo cytokinesis, which is the division of the cell membrane that forms 2 daughter cells. The reason why cells must divide into 2 rather than continue physical growth is because the larger the cell, the less surface to volume ratio it has, meaning that it will be less energetically efficient to carry out biological processes. Dividing into 2 cells helps to resolve this issue. So in the next few slides, I will demonstrate the cell cycle, and I want to introduce the important components involved. Many organelles are not shown here for the sake of simplicity. The plasma membrane, which is the border of the cell, is represented by this tannish-colored circle. The purple-colored circle is the nuclear membrane. The nucleolus is the dark purple circle. It's not really necessary to the process of cellular division, but it's still shown because it's part of the nucleus. You also have chromatin. Chromatin is going to be represented by these squiggly lines you see here inside of the nucleus. And finally, centrioles, represented by these orange rectangles. These produce spindle fibers, which are used to help to divide the chromosomes to each potential daughter cell and help split the cell. During interphase, the cell undergoes normal growth processes while preparing to divide into 2 daughter cells. Interphase is broken down into 3 stages, G1, S, and G2. Cells must successfully move through these stages in order to move on to mitosis. The G1 phase, or first gap phase, is called such because at the molecular level, the cell is preparing the DNA to be replicated and making excess ATP to prepare for replication. You might have also noticed that it physically begins to grow. The S phase, also known as the synthesis stage, is where the DNA is duplicated. Right now, the DNA is organized into chromatin, which is less condensed in chromosomes. When S phase is complete, there are 2 copies of DNA, which will eventually become sister chromatids in prophase. This is also where the centrosomes, which are important components of the cytoskeleton, start to replicate. To help prepare for division of a cell into sister chromatids, centrosomes give rise to the mitotic spindle, which will help the cell divide. The G2 stage occurs after the S stage. Here the cell must replenish any lost ATP needed to replicate DNA, so now that it is prepared for mitosis and cellular division. Also, you'll notice organelles become duplicated and the cell continues to grow. Before the cell can enter mitosis, it must pass some checkpoints related to proper DNA replication and ensuring that the cell has the proper resources to continue on to mitosis. If everything's good to go, then the cell enters the mitotic phase. If not, the cell will enter a period of stasis or be destroyed, but we'll look at that later. The mitotic phase is split into 2 parts and 5 phases. Karyokinesis takes place in 3 phases: prophase, prometaphase, and metaphase. The purpose of karyokinesis is to divide DNA in the original nucleus. Cytokinesis takes place in 2 phases, anaphase and telophase. The purpose of cytokinesis is to split the cells into 2. The first stage of mitosis is prophase. During prophase, chromosomes condense and first become visible under a microscope. The nuclear envelope disintegrates into small vesicles, and membranous organelles like the Golgi complex and endoplasmic reticulum will start fragmenting towards the edge of the cell. The centrosomes begin to move towards the opposite ends of the cell. Then it moves on to prometaphase, where more disintegrating of the nucleus occurs and the mitotic spindle further develops. Chromosomes also become more condensed with sister chromatids, the centromere or area where the chromatids attach. The mitotic spindle is made up of microtubules called spindle fibers, and there's 2 types. Polar microtubules, their job is to push the poles of the cell away from each other during mitosis. And kinetochore microtubules will pull the chromosome, the sister chromatids, away from each other and towards the poles during mitosis. During prometaphase and metaphase, the formation of the mitotic spindle is complete. Motor proteins on the kinetochore microtubules pull each chromosome in opposite directions, causing the chromosomes to line up in the middle of the cell. The imaginary plane formed by this is called the metaphase plate. At the end of metaphase, karyokinesis ends and cytokinesis begins, which then leads us to anaphase. And during anaphase, centromeres split and sister chromatids are pulled by the spindle fibers toward opposite poles of the cell. Replicated chromosomes split into 2 identical sets of unreplicated chromosomes, and as soon as they are no longer attached at the centromere, sister chromatids become daughter chromosomes. In addition, motor proteins of the polar microtubules push the 2 poles of the cell away from each other. So next is telophase. During telophase, a new nuclear envelope begins to form around each set of chromosomes, and the mitotic spindle begins to disintegrate. Chromosomes then begin to decompress back into chromatin. When 2 independent nuclei have formed, mitosis is complete. Now we must complete the division of the cell. So cytokinesis typically occurs immediately after mitosis. During this process, the cytoplasm divides to form 2 daughter cells, each with its own nucleus and complete set of organelles. Remember, cytokinesis actually begins in anaphase and it completes after telophase occurs. Cytokinesis in animals, fungi, and slime molds occur when a ring of actin and myosin filaments contract inside the cell membrane, and what it does is it causes itself to pinch inward, forming a cleavage furrow. This causes the cells to divide into 2. Cytokinesis in plants is going to be slightly different, whereas the other stages of the cell cycle are pretty much the same as animals. In plants, because they have a cell wall, the way it divides will be a little bit different. So cytokinesis in plants occurs as vesicles are transported from the Golgi apparatus to the middle of the dividing cell. These vesicles fuse to form a cell plate. Thus far, I've given the impression that all of our cells are constantly dividing. While this is true for skin and blood cells, most tissues in our body are regulated or have cell division stop altogether once new cells are formed. Any cell that is not actively preparing to divide during interphase is said to be in the G0 stage. Sometimes the G0 stage is temporary due to the conditions of the organism, And in the case of mature muscle and nervous system cells, it is permanent. This explains why nervous and muscular disorders are really hard to treat. The cell cycle length varies from species to species and within the developmental stages of each species. Embryos and younger organisms undergo the cell cycle much faster than adults. Regardless, the cell cycle is tightly controlled by both internal and external mechanisms. Since mitosis and cytokinesis are a form of asexual reproduction, all cells must be exact duplicates of their predecessors. Otherwise, a deadly mutation could form and proliferate to other cells around the body. Not all mutations are bad, but cells have mechanisms that prevent them from happening. Some of these mutations can lead to cancer, which we will look at later. Thus, biologists have observed 3 essential regulatory checkpoints to ensure that the cell cycle produces exact duplicates and prevents abnormal daughter cells as well as cancer. The first checkpoint occurs at the end of the G1 interphase. This is also the most important, as the cell will either be put in the G0 state or continue with the cell cycle. The G1 stage is all about physical growth of the cell. Cells at this point need enough resources to continue physical growth, and the DNA in the nucleus is undamaged. If these conditions are not met, then the cell enters the G0 stage indefinitely, or it is destroyed by the immune system. Towards the end of interphase and before beginning mitosis, cell size and resources for cell division must be adequate before continuing. Perhaps the most important check is to make sure that the DNA is undamaged and was appropriately replicated. Otherwise, the cell will not undergo mitosis. There are a lot of moving parts in the cell cycle. Therefore, the last checkpoint occurs after metaphase. This is the spindle checkpoint, which ensures that a spindle fiber is attached to each chromosome. Failure to ensure that each mitotic spindle attaches to each appropriate chromosome will cause abnormal cells. In mammals, the immune system generally wipes out deformed cells in order to prevent cancer, which is a topic of our next video alongside the prokaryotic cell division.